Session 2 is cell-mediated immune responses to SARS-CoV-2. So we've got two fantastic talks in this session. Our first speaker is Bruce McLachlan from Cardiff University. Bruce is a structural immunology research associate at Cardiff University, and his work normally applies structural biology and biophysical techniques to gain insight into immune function. His talk today is entitled Using Structural Biology to Define Model HLA Class II Presented Peptide Antigens of SARS-CoV-2. Our second speaker is Professor Andrea Cooper from the University of Leicester. So Andrea is a Professor of Cellular Immunology at the University of Leicester. She received her undergraduate degree from University College London and doctoral degree from the London School of Hygiene and Tropical Medicine in the UK. After her PhD, she moved to the US and spent many years there before moving back to the University of Leicester. Her talk today, and actually I haven't got your talk, Grace, I have it on the slide, so peptide-specific stimulation of low-volume PBMCs in a broad healthcare worker cohort. So Bruce, I'll hand over to you for your presentation. Thank you.
Sorry, 1 second. Is it gone? Just having, it was all fine until I had to do the important bit. Here we go.
That always happens, it's fine.
Yeah. There it is. Brill, can you see that okay?
Yeah, that's great, thank you.
Okay, so thanks very much for the invitation to talk today. So my name is Bruce McLachlan, so I'm in the Division of Infection Immunity in Professor Alwyn Gallimore and Professor Andy Godkin's lab. As Lisa said, I'm a structural biologist really by training, and today I'm going to be talking about how we are using structural biology to define model HLA class II presented peptide antigens of SARS-CoV-2. So I'm particularly interested in CD4 T cell responses and how HLA class II molecules present peptides to these immune cells, sort of in the context of both tumor antigens and in the context of viral antigens. And I wanted to put this slide up here to show in sort of the general process of processing and presenting HLA class II peptides to highlight a couple of key features that we're particularly interested in about this system. So as we know, viruses uptaken by professional antigen presenting cells, and these proteins are digested into short peptides where they are then loaded onto HLA class II molecules in the MSC class II compartment, where if they have sufficient affinity to bind into the groove and displace the clip peptide in a process that's catalyzed through HLA-DM, these peptides are loaded onto the HLA class II molecule and transported to the cell surface for interrogation by the T cell receptor repertoire. And on the right here, just wanted to show what we think is a really key feature about HLA class II peptides, and that is that the peptide binding groove of HLA class II molecules is quite different from class I, and it has what's called an open-ended peptide binding groove. And this means that class II molecules bind a 9 amino acid core region that is then flanked by other residues that are termed the peptide flanking residues, or PFRs. And we have data, and we're really interested in showing that these PFRs are much more important than maybe first appreciated, because we see that they are, in fact, whilst not part of the core binding region, they are also important for both creating these interaction networks between the HLA and the peptide, and also the recognizing T cell receptor. So the outcome of this is that for HLA class II presented peptides, unlike in class I, so in class II, what's actually presented is a large array of peptides that are all based around the same core region. And then these peptides, which are called nested sets, have a different array of abundances when eluted from HLA class II molecules on the cell surface. So over many years, X-ray crystallography has been used to help drive mechanistic insight into this system and how T cell receptors recognize these peptides presented on HLA class 2. And we know that the T-cell receptor in general binds at this diagonal angle across the peptide binding groove, interacting with both the peptide, which is held here in the peptide binding groove, as well as the HLA class 2 surface. And one of the ways that we use crystallography is to define HLA class 2 presented peptides and to find them and generate model antigens for the system. So the different HLA alleles all have different specificities for different peptides. And we can see that when we look at the structures, we find, for example, shown here, this is the PKY peptide, which is a DR1 peptide and also binds to DR4. It has this characteristic tyrosine at position 1. And this is indicative of what's shown in here. We have these motifs for each different allele of HLA-DR. So I mentioned that we're interested in these peptide flanking residues, and what's really interesting is when we solve structures of HLA-DR1, when it's the peptide cargo that it's holding is much longer peptide, in this case a 20-mer, we find that these peptide flanking residues are actually really important for what constitutes that peptide as being an antigen. So looking as far as P-3, so that is 3 residues into the N-terminal flanking residue, we see a really lot of contacts that are happening between the peptides and the HLA. So these are really important contacts that are contributing to the immunogenicity of that peptide and its display on the surface on HLA class 2 molecules. So back in November, we set out to try and use our crystallography toolkit to try and define and characterize epitopes from SARS-CoV-2 in the context of HLA-DR1, because from a practical point of view, it's an allele that we may be able to make relatively easily, and also it's a commonly occurring allele in the population. And we wanted not just to focus on spike, we wanted to try and take a more general approach and identify non-spike epitopes as well. So with this in mind, in around November time, there was quite a few studies, many from Alessandro Sept and Alba Grafoni's lab in the States, and also Tara Dong's lab in Oxford. We took a look at these papers and we looked through the lists and the supplementary materials and tried to collate a list of epitopes that were likely to be restricted to HLA-DR1 and also DR4 for other studies, and filtered these. In many of the cases, the restrictions hadn't been fully elucidated, but were known to be in individuals who contained these or who had these alleles. So we filtered the peptides that we found based on their predicted binding to DR1 or DR4, and registered these peptides as 15-mers centered upon the 9 amino acid core shown here by the underlining. And then once these peptides arrived, we first wanted to actually run, being so dependent on the peptide binding algorithms, wanted to generate experimental data to show whether these peptides were actually able to bind to HLA-DR1. So to do this, we have a peptide binding assay, which is based on competitive inhibition. So I set up a peptide exchange reaction where in the well, we have our HLA-DR molecule, which we've made in E. coli, which I'll go on to talk about how we do that. Also in the well, we have a surrogate marker peptide, which is the CLIP peptide, which is biotinylated at the N-terminus. And then we introduce a titrating amount of our candidate peptide that we want to test whether it's a good binder or not. We incubate that at 37 degrees overnight at pH 5. And if this peptide, the candidate peptide, is able to enter the DR1 binding groove, we then set up a strip HRP reaction where displacement of the marker peptide by high affinity candidate peptides will result in a loss of signal and an inhibition. And from that, we get a binding curve, an example shown here for the PKY peptide that I mentioned earlier, which is from influenza A virus. So based on that, I was able to characterize the panel of peptides that we'd selected from the literature and filter them down to peptides that we were able to experimentally show were able to bind to HLA-DR1 in our assay. What I guess this first highlighted was that whilst we had quite a few peptides that were, well, in fact, most of the peptides were predicted to bind well to HLA-DR1, we didn't quite see this experimentally, but we were able to identify 3 peptides which found just as well as, or with comparable binding to our sort of model PKY peptide from influenza A. So from this, we selected these peptides to take forward for further study. And this is through our crystallography pipeline. So in order to do the structural biology experiments, you need to first make your protein. For HLA-DR, we do this using an E. coli expression system. So we express the alpha chain of DR and the beta chain of DR separately in E. coli and purify the synthesized protein as inclusion bodies. We then have to refold that protein by putting 1-to-1 molar ratio of both chains into a refold mixture in the presence of our peptide of interest. We leave that over the weekend and hopefully it will form our native HLA peptide complex. We then diafiltrate the sample and purify it by anion exchange, affinity chromatography and side exclusion chromatography. We then have to assess the purity of our protein using the SDS page. And then if it's deemed to be pure enough, we will take it forward for crystal screening to obtain samples that we can then take to diamond light source to fire with X-rays and collect structural data on these proteins. So here's an example of those 3 peptides that I mentioned before. So one from the membrane protein, one from NSP3, and one from NSP14. And example of the protein at the end of the whole process and the purity that's obtained. So based on these 3 peptides, I should say that this is still very much a work in progress. We've only recently collected this data and we're still trying to analyze it and put it all together. So far, we've been able to crystallize and collect nice data on 2 of these peptides. I'm still working on this peptide here to try and complete the 3 in the Sept. And what we are starting to learn from these structures, so starting with this KSA peptide from NSP3, we see that the register in which the peptide binding algorithms predicted was in fact the register that we found were held within the peptide binding groove, and it does adopt a very classical HLA-DR1 binding motif for the hydrophobic residue at P1, aliphatic residues at 4 and 6, and then a slightly less common serine at position 9. This epitope isn't very well conserved at all outside of SARS-CoV-2 when we look at other coronavirus family sequences. And again, we're working to elucidate a bit more insights into what we can see in these structures in terms of the contribution of each residue to anchoring within the HLA binding groove and finding out why this peptide binds so well to HLA-DR1. It's quite a similar story with the second peptide that we have structure for, so LDA from NSP14. Again, it was bound in the same register that was predicted with a very similar motif, but instead we have a phenylalanine at position 9, which is not typically seen at P9, but yet with our experimental data, we can show that it's a very good binder to DR1. This peptide is much more highly conserved across different coronaviruses. So it would be interesting to see whether any of these other variants might actually also bind to DR1 and offer some sort of cross presentation across different coronaviruses. So working with another postdoc in the lab, Chen, who has started to try and elucidate our own data on whether looking at the immunogenicity of these peptides in DR1 donors. So these are just local donors. She's looked in 3 donors so far and has found in cultured ELISpots and interferon gamma responses that the peptide 24 seems to have quite a nice response across 3 donors checked. The peptide 9, which is this one here, has come up in one of the 3 donors, and our other peptide, peptide 22, so far isn't quite so immunogenic as yet. So Chen is working to grow T-cell lines against these epitopes so that we can hopefully extract T-cells and TCRs from these individuals. And then lastly, one other thing I'd like to talk about. So we have been collaborating with Heather Long from the University of Birmingham, who's been working hard to map epitopes of CD4 T cells in a number of different individuals expressing different alleles of HLA-DR. And she's shared with us some of her data where she's found 2 epitopes that are HLA-DR1 restricted, where she's grown clones out of these individuals who have been infected, and using a library of overlapping peptides, elucidated the optimal peptide for each of these 2 epitopes that are both contained within the spike protein. So we have been working to try and see whether we can use our crystallography pipeline to help drive mechanistic insight into how these peptides are being presented. Over the last couple of weeks, we've just been able to express and crystallize these 2 peptide HLAs, but I've not yet been able to take them to the synchrotron to collect the data yet. But we are really excited to see the structure of these peptide HLAs, particularly as they weren't particularly well predicted to be good binders to HLA-DR1, yet from Heather's data, we can see that they're quite immunogenic. So it'd be really interesting to see what the register and what the binding motifs are for these peptide epitopes. So with that, I'd like to thank my PIs, Andy Godkin and Alan Gallimore, and everyone in the lab who's been involved. Andy Sewell and his lab, who helped we work in amongst. Pierre, who runs the crystallography activities at Cardiff, Heather at University of Birmingham and her team, Diamond, who for access to the synchrotron, and the Welsh Government, who provided some funding for this research. Thanks.
Fantastic. Thank you very much, Bruce. So we'll take questions at the end of the session. So if Andrea's happy to do her presentation now, share your screen, if it works.
Are you seeing my screen?
Yes, we are. Don't know if you want to put it onto slideshow, then we have the whole
I will
screen.
do.
Yeah, fantastic.
Yeah, I just wanted to make sure we're in the right. Okay, everybody.
Yeah, brilliant.
Excellent. Well, again, thank you very much, everybody, and I'm delighted to be at this meeting. We've been so. I am a mouse immunologist, just first and foremost, just to let you know that. But I've been interested in examining peptide-specific responses in humans and sort of tying that back to some of the work that we've been doing in animal models for a long time. So we, of course, have had an opportunity in the last year to expand what we'd been doing in tuberculosis into the COVID issue. And I've got, so what we're going to talk about here is peptide-specific responses in a broad healthcare worker cohort. And what does that mean? I just wanted to highlight the person who's been responsible for this cohort, and this is Dr. Manish Parikh, and he generated this UK REACH cohort, which is a very large cohort of healthcare workers. And then within that cohort, we've got our a subset of determining the immune response in ethnic minority healthcare workers to COVID-19. And we've got the word infection there. We started this up probably end of sort of mid-2020, sort of March, April 2020. And of course, when we started it up, we thought we were going to do infection, but in fact, by the time we got organized, we were really addressing the immune responses to vaccine more than anything else. So this is the cohort that we're talking about. And what is that What is that cohort? So the recruitment commenced about March 2021. So far, we have 480 healthcare workers recruited. The majority of the participants have been vaccinated, and 30% have an episode of confirmed or suspected COVID-19. And of course, being healthcare workers, their healthcare records are accessible for us, and they are quite thorough. Now, the interesting and important part about this cohort is that it's 37% of ethnic minority groups, and so that's really a much broader coverage than normally. And so 400, so that's sort of one of the key elements of the work that we're doing. So you can see that we've got a we start out with a complete baseline questionnaire. So we're really covering a lot of parameters here: gender, age, ethnicity, job role, which we're equating to exposure, living arrangements, again, exposure, and health, general health, including BMI, and then infection and vaccination status. So we're quite confident in those roles. And then we've taken a baseline blood, and that's basically the samples that we've got so far. And then between the now and the end of the year, there'll be, hopefully we will have boosters given to those individuals, and we'll take more blood then. And then February, March of next year, we will have our final sample of blood. And so what we'll have is a selection of activities going on. And these samples that we're taking, they're in the context of other work going on. If you look at the bottom of the slide here, we'll be doing a T-cell response in our lab. That the same fresh blood will go down to Oxford Immunotech, and in fact has gone down for the Spot assay. We will have antibody responses analysed by the PHE. We will be doing proteomic analysis on these same samples, and this is by Ralser at the Crick, and also in a subset, in a deeper analysis here at UOL. And we'll also have samples taken and stored for genetic analysis. So it's really a really large cohort. It's quite diverse. There's a lot of samples being taken. And in that context, we wanted to look at the T-cell response. So what have we done? And I think this is probably the naivety of an animal modeler going into the human system. So we took blood and we thought, I'll try and get as much as I could. I got 15 mils blood total. So 2 7.5 mil EDTA tubes. We took those tubes and did peripheral blood analysis, peripheral blood purification. So we've got about 4 tubes with about 10 to the 6 cells per tube. And then we wanted to do a cell stimulation, follow that through with flow cytometry and analysis. And we didn't know how many we would be able to do, but the first thing we wanted to think about was what about the thing that would drive the T cells and allow us to identify the responses in these individuals? Now, this is where my animal modeling work comes in. And I've been working on tuberculosis for a long time, and my understanding of how T cells behave is based on the concept that if you look at this picture at the lower left here, you've got, let me just make a little pointer. So if you look here, you can see that the green is the bacteria, the blue are the macrophages, and the red are the T cells. So everything I think about T cells is how they work in the inflammatory environment. And what we know from our animal modeling is that antigen-specific T cells behave and are very, very different in terms of what they're doing and how they're surviving. So this is just an example. We just did we added basically 2 lots of cells together into one environment and asked the question, if they're antigen specific, how do they behave? And it's actually to do with whether they've got a cytokine receptor on their surface or not. But the key thing is, if it's just regular CD4 cells or regular non-antigen specific cells, they basically behave identically within the environment. But antigen specific cells, as identified here by tetramer, are behaving very differently. And in fact, what this data shows is that cells that lack a specific receptor are much more fit to function and persist within an inflamed environment. And so the idea for us is, and the message that I just want from this slide is that if we're not looking at antigen-specific cells, but just looking at total, and in fact, if you just look at total CD44 activated cells, the effect is not very strong. We can't see a strong effect. But if we're looking at antigen-specific cells, then that effect is much more pronounced, and we can see a really strong signal. And I think I'm probably preaching to the converted here. If we don't look at if we're looking at a broad response, and I think this comes back to Bruce's talk, we've got to know what those specific peptides that are being recognized and how are they mediating their effect on the T cell. And some other work that we've been doing in terms of peptide-driven vaccination tells us that it's critical. The nature of that interaction with the HLA is critical in the type of T cell that's induced and how that T cell then behaves when it sees the natural ligand. And so I was just very excited to see Bruce's talk and just understanding the importance of this initial interaction between the T cell and its peptide. So that's a long story. And so when the pandemic started, we were interested and we knew we were going to get peripheral blood cells. And so I reached out to Alex Sept. We've been working with him and Cecilia Arlatham in La Jolla, looking at TB T cell responses. And so I said to him, we'd like to, and in fact, this is work from Daniela Weisskopf, where and that's that Mateus paper you've probably seen, where they were looking at antigen-specific responses to circulating SARS-2 circulating coronaviruses and seeing how those influence the behavior of T cells and how those T cells looked, and compared those to specific SARS-2 specific peptides. So they had a set of peptides. I wrote to them and said, let's look at this in our population here. But of course, by the time we got everything sorted, most of our population were vaccinated. So our incidence of disease reduced. So we then communicated again and said, okay, well, what should we look at now? So we decided that what we were interested in was comparing spike protein, which is of course part of the vaccine-induced response, and non-spike protein, which would give us an indication of naturally induced, infection-induced T cell responses. So, and I just wanted to show this picture here. This is the bulk unstimulated bulk cells, and this is the nature of those cells there and how they look in terms of markers on their surface. CCR7, so central memory, naive, effector memory, and with CD45RA right there. So you can see those populations in that bulk population. If we look at the cells that are stimulated by the CD4, and this is a pool of peptides, so we're just trying to cover every base here. It's a little bit non-specific, but we're covering every possible. They selected these to be able to be useful worldwide in terms of their ability to drive human T cells. So in this pool, we can see there's a selection of cells that are activated and by this pool of peptides that are associated with infection, and you can see that a lot of those are effector memory. And then in the spike protein, we've got a large majority of cells, and this is in blue here on the background of the total population. You can see a lot of effector memory cells there. So we said, okay, so when we think about what it is that we're going to stimulate our cells with, and again, being naive and rather enthusiastic, we said, okay, we'll take our tubes and we'll see if we can stimulate them with the spike pool, multi-pool, and then this, what they called CD4 remainder, really. But these are basically peptides that are associated with natural infection, and these are peptides that will be driven by the vaccines. And then, of course, we've got a positive control there. And so we stimulate them, and we also added in a couple of extra steps here, and I'll just talk about that in a minute. And so here we've got just our general two things we wanted to do. So again, naive and somewhat brave. We've got obviously a live dead selection and a dump channel, allowing us to get single cells which were positive for CD3 and CD4. And we would then, so when we cultured our cells, we just put proveldin in the culture for the last 2 hours, and we're looking then at TNF production. So looking at TNF production, we then would look at the interferon gamma and the IL-2 responses, and then also just looking directly at IL-2 responses in our CD3, CD4 positive cells total, and interferon gamma responses total, and then taking those interferon gamma and looking at our HLA-DR. And these are just sort of fairly standard, and there's a lot of work going on now in looking at these kind of markers in COVID specific responses. And there's a lot of literature, but I think what we want to do is look at that literature, look at these cells in our large cohort, and take this kind of information forwards. So we think we can get quite a lot of this. This is obviously driven by SEB here. We also wanted to look at activation markers. So activation-induced markers is quite a common tool. I think it's used quite widely, and there's a variety of markers that people are interested in. Here we decided, we thought we'd just focus on, again, we go for our CD3. And so just to say, this for the intracellular cytokines, it's a 6-hour culture with 2 hours of proveldin at the end. For the activation markers, it's a 24-hour culture. So 2 different ideas, really. In the intracellular cytokine is, what are the cells ready to make? And in this 24-hour culture, it's what are the cells able to develop and become? And sort of thinking about those markers, are they being able to be induced by activation to change their expression and be responsive? And so that's sort of 2 different things to be undertaken. So this is just, we take our CD3 cells, CD3, CD4 positive cells, look at CCR7 and CD45RA, and then look at their expression of things like PDL1 and CD25, OX40 and CD25 in the different populations. So you can see that's a nice sort of simple, straightforward tool to look at a lot of different parameters of these T cells. And again, the problem for us really is the fact that this is SCB data sets here. And so we thought, well, how are we going to optimize this study? So originally we wanted to use fresh cells, and we could have done that. We could have done fresh cell cultures and then frozen the cells and then done the flow together in a batch analysis. But we thought perhaps we would just see if we can get away with what's the difference between the fresh and the frozen cells. And you can see, whilst there's a little bit more dynamic range with the fresh cells, we do get a sizable population, a positive cytokine producing cells. So we kind of, and we did the same for our IL-2. Again, the dynamic range is not quite as good, but we do have a positive population. So for ease, we decided to move forward with frozen cells. I think that's because we were also constrained somewhat by access at this time. So we also, critically, of course, we've got very, very low frequency of responding cells when we look at our peptide-specific responses, and therefore we just made sure that we've got our FMOs solidly there for everything. And just to show you here, we've got our TNF and our IL-2 and interferon FMOs. We can make our gates. We feel quite confident in these populations, even though they're quite small. So nothing surprising there. What we had originally done was not actually use anti-CD28 and anti-CD49D. And so we thought, well, let's see how that works. And then we put those in, and we were getting much more robust responses there. And in fact, and I feel bad because the student is actually doing the analyses right now of our first set of data from our selected patients, or not patients, but subjects. And so this is a data set I can just show you here, where we've been using CD28 and the CD49D alongside of the CD4 spike protein in one of our subjects. And you can see that we've got here a population of cells that are responding in terms of IL-2 signal there. And again, comparing that to our activated FMO, it's a fairly nice population. I think we probably move those gates up there a little bit. It's a little bit, again, we've got to be confident in our signal. It's a low frequency signal, but I think it's really going to be very interesting coordinating that signal alongside of all of the other parameters that we've got in our cohort. And again, this is the same set of data. So with there's our FMO gate, and there's our CD4 spike pool with stimulated with anti-CD28 and CD49D. Again, frequency is low. But we've got this is from one tube. We're actually going to run 2 tubes in our when we're running our full samples, and I think we've got a fairly robust signal that's positive. And so we're moving forward. It's taken us a while to sort of, we didn't want to just jump in and try, especially with the low frequency and the low number of cells that we had. So, and thinking about it, we're going to have about 2,000 samples. So and for every sample that we have the T-Spot assay for that sample, and that's the data set that we've been collecting in real time. And so because this is not my data set to be given away freely, I've just called these, this is just a set of data that we've plotted, and this is the T-Spot assay. You can see this is the number of T-spots, and this is done with Oxford Immunotech, Andrea at Oxford Immunotech. And so what we've got is a set of data that shows us for these 4 different sets, which could be, you could range them by ethnicity, you could range them by age, you could range them by distance from time from vaccination, a variety of different kinds of parameters. Looking at it, we can see a range, and we can see, thinking about the spike proteins, we can use this data, and that's what we're going to do, use this data to focus on the groupings that we have for our, when we do our flow cytometry analysis in depth. And so what they've been doing is basically the clinical people, and I'll sort of name them in a minute. So what we've been doing is basically saying we can run about 8 samples per sort of stimulus, and we want to ensure good comparison capacity. So we're going to basically have our clinical colleagues identify the RD numbers based on those T-Spot assays, and then link in and then basically do those 8 together. And we can run about 3 sets of 8 every week if I want to keep my student from not sort of complaining too much. So we can get through those 2,000 samples, where in each sample you've got a good comparator between, you've got basically 2 of each set that you're interested in. So it looks about 80 weeks, but I think we can crank that up the more exciting the data gets. So all that being said, so this, in fact, I just want to say that's the data you're seeing about COVID, which is not a lot of data, but just to say that it is feasible. We do have these responses. We will be analyzing them in full. And just to sort of reach back and say why we've been so focused on doing this is because of the strength of this cohort. And so what we have in the green box here is this is sort of a broad survey of all of our cohort members, all of our almost 500 healthcare workers. We're going to look at all of those in the first instance. In this second instance, we're going to have pre and post vaccine boost, and so those samples then we'll be able to work on depending on how what data we see here. We will go through and look at them all, obviously, but that's a very powerful tool to sort of focus this response here. And then we will also, and that will allow us to think about the effect of boosting, the effect of ethnicity, the effect of BMI, the effect of exposure rates, and the nature of their underlying immunoglobulin response, their underlying T-cell response, and their underlying initial response to vaccine. So a lot of potent information there. And then, of course, this final collection we'll look at the longevity of the response. And we've also got a subset of the study that we're going to think about its interaction with flu vaccines. So there will be a subset of people who will be also at the same time getting the vaccine. We can look at their influenza-related immune responses if we've got enough blood. So just acknowledgements here. Alex and Daniela were very helpful, very supportive, and communicate really very rapidly with us. I don't think they ever sleep, to be honest. I think I'd like to really point out Amar Jarki, who's the PhD student who's doing this work. He's really very well doing the cell culture, developing the flow cytometry, ensuring that we're ready to go. And I think he's in the audience, I hope, but he may be on the flow cytometer collecting the data from the first study that we've done. And I'd like to also make a shout out for Chris Martin, who is the clinical fellow. He's been doing a lot of the clinical recruitment, but also the analysis of the impact of different parameters on that T-spot response, and also generating the clinical groups that we will put through the assay once we start to work. And of course, Andrea Tattersall from Oxford Immunotech, Claire Hood is the research manager for this, and obviously it's a massive coordinated event, and Manish Parikh, he's chief investigator at UK Reach and direct, and has ensured that the MRC has been supporting us. And obviously I'd like to thank the MRC for their willingness to take that leap of faith and this large cohort and the immunology detection in this large cohort. Thank you very much.
Great, thank you very much. Thank you.
Should I stop sharing?
Yes, please, if that's okay. Great, thank you. So at the moment, there's no questions in the Q&A chat. If anyone has got any questions, please feel free to post. But I'll start. I've obviously, as we've been talking, writing down lots of questions. So I'll start, Bruce, with you, if that's okay. A couple of questions. So obviously CD4 T cells are really important, but we also know with SARS-CoV-2 infection that we need a good CD8 T cell response. So have you got any plans, or have you started looking at any of the peptide binding on MHC class 1 as well as looking at the MHC class 2 molecules?
So we're focusing on MHC class 2. There is a lot of work going on in the class 1 space. So there's quite a lot of competition there. There's one epitope in particular that's restricted by HLA-A2 from the spike called YLQ. And there's been some nice studies to show, crystallography studies to show how this peptide is recognized, and any variants of that peptide in the databases that might be affected by variation and affecting the response in A2 individuals.
Okay, Danny, I'll hand over to you as you've raised your hand.
Yeah, can you hear me?
Yes.
Yeah. So I was going to kind of try and start a row, if I could, between Andrea and Bruce, in a sort of immune philosophical sense, that if you took, all the lovely work from both of them, and if you took everything that Bruce says about, the sort of optimal positioning of sort of peptide in the groove and everything, and then you take Andrea taking a sort of Alex Sete style approach, which is something that I've bickered with him about probably for the last 20 years annually at immunoepitope discovery meetings. Andrea's kind of doing a kind of mega competition assay for the peptide binding groove, for thousands and thousands of peptides, some of which are relevant and some aren't. On the one hand, you could say, and Alex would say, and Andrew would probably say, the proof of the pudding is in the eating, and if a response comes out the other end, then we're happy, which is probably quite a good thing to say. But it is a kind of enormous competitive binding assay. Discuss.
So I think yes, it is a black box in between what we put the peptides in, and we wait and see what the response is. And we're hoping that in fact that the affinity for the specific peptide for the HLA will be sufficient that will allow us to get a response. I think it is the tool that we can use, and that's those caveats, as you say, are part of the analysis.
Yeah.
Can I sort of add related to that? Oh, actually, Bruce, I'll let you answer first. Sorry.
Yeah, just from my point of view, we to do this work, we kind of need to be working with the best peptides, and the place that we can start is by first experimentally showing that this peptide is in fact a good binder, because producing the DR without a good peptide, it's much less stable and it falls apart. So from an experimental point of view, that's why we need to work with at least the best binders, and then appreciate that that's not necessarily definitely means that they are going to be the ones that generate the most potent immune response. But that's why we also need to work together from both angles.
Absolutely, and sort of related to that, Andrea, I was quite surprised that you're also adding in CD28 and CD49 to try and push your cells. Is that because the peptides that you're using aren't sufficiently driving that immune response, or do you think it might be a numbers game? Because obviously you're only getting relatively small blood samples, and I know from the work that we've been doing in Nottingham, you need a lot of cells because you're only looking at very small populations of cells of interest. So yeah, I just want your thoughts on the addition of those extra stimulators in your assay.
Yeah, so when we didn't have them in, so this is a good question. What is it that we don't see sufficient to actually see anything? So what are we seeing when we add those in? And so I think we're looking at, that's where our numbers and our background information, and then also what we see with our boost response will be because we'll expect to see a much larger response when we've got our vaccine boost occurring.
I don't know if you put the data up, and I might have missed it, and apologies if I did, but if you've done it where you haven't added the spike, but you've just the same sample added 28 and 49.
Yes. So
and
that's
what's the response there? Is that a zero response?
Yeah. So that basically is the FMO response. We have, yes, we have the peptide. Sorry, you're asking for CD28.
I'm asking for the additional stimulations. Are you doing any with just the additional without?
That's the negative control. So that all the wells will have those in, and then the spike peptides, then anything over and above. Yeah. So we would be looking that would discard any cells that were ready to go for no other reason than those co-stimulators in there.
I think all of us working in the T-cell arena is really conscious of the fact that we're working with very small numbers of cells, trying to identify populations and try and generate some meaningful data out of it. So yeah, I think. Sorry. Isn't
that the most exciting thing that we've got right now? Because we've never had this number of samples, never had the integration that we've got, which is so superb, that people are doing very similar, and we can backtrack everything. I mean, that's, to me, that's why I took the punt on doing the work that we're doing now, because I thought, well, it's going to be in the context of a broad swathe of very good clinical data.
Yeah. I think as well in Nottingham, we of course have got the ACE cohort, and I think I might be a step in later one because I think we've got a speaker missing. So we're doing very similar sorts of work to you, but in our student populations who have had asymptomatic COVID-19. So yeah, we can continue the discussion about the assays and how to develop them and how to get the most out of the data later. But I think really it sort of goes back to you, Bruce. So I mean, one of these peptides or one of these sequences going to be available to the scientific community to be able to use in our assay systems?
Well, we want to try and complete the set and get as many structures as we can. And then obviously once the structures get published, they go into the database, and then they're freely available for everyone to interrogate on their own.
Fantastic. Okay. Are there any more questions from the audience? Yes. Adrian.
Just a quick question for Andrea. Is infection, reinfection, or infection despite vaccination a hard endpoint in your study? And what was the sort of baseline seroprevalence or rates of infection across your healthcare worker cohorts?
So we have that data set. So we do have, but as I said, in that first set of data we're looking at, it's a broad exploratory analysis, but we do have, we have their seropositivity because they've all had, and we do have their case rates as well. And we will know if they get, again, so everybody's they do have a regular questionnaire as well for symptoms. And so that's again, it's an exploratory undertaking, and we're hoping for enough numbers, but it's not an endpoint as such.
Andrea, sorry, it's asking another one to you again, but you mentioned non-spike protein that you used. What is your non-spike protein? So you said spike protein, but what's the non-spike protein?
So they call it, and so you'll have to forgive my ignorance. This is Alex's and Daniela's reserve, or what they call it, remainder. So it's it's essentially a set of peptides that they were using. And when I originally spoke to them, I selected the peptides that were not recognized by other coronavirus. They weren't stimulated by other coronaviruses. So it's basically COVID-specific non-spike peptide pool. So we wanted it as broad as possible to try and get that an evidence of infection, but not vaccination. Vaccination would be and infection would both be stimulated by the spike, whereas the remainder would just be an evidence of people who had been infected.
And is that cross-reactive with seasonal coronaviruses? Because I know a lot of the work.
No, that was the point about them. They are
non-cross-reactive.
And the paper that that I first sort of approached them about was because they were differentiating between the seasonal and non-seasonal.
Okay, great, thank you. That's really useful to know. And Bruce, I'll come back to you with one final question, if that's okay. So I was quite surprised when you said that the predicted didn't actually match your experimental data. So why do you think that is? And is that normal for most sort of, I don't know what the sort of normal predicted matched up to biological data actually is. So is it sort of the normal levels you're expecting, or is this like a really unusual situation?
I'm not quite sure how normal it'd be. One thing that is quite important is that if we miss the core binding region of a peptide, so a peptide might be immunogenic, and then it's predicted to have this core binding region, and then if we synthesize that peptide and actually it wasn't in there, it was actually 3 or 4 amino acids to the right, then we might miss the peptide. So it's important, to get the core correct. The prediction algorithms are pretty good. But I guess just once you start doing large scale, scans across the whole genome, there's going to be some that are correct and some that aren't. And actually, if looking at the two structures that we have, they both, as I said, have at the P9 position, they have residues that aren't typically seen in the motifs, but they both have a serine there at P9. So those are the kind of things that would be downscoring them in the predictions, but experimentally we can show that they're actually very good binders.
Okay, fantastic. Yes.
So so Bruce, this is probably just again, it's a little bit of a hypothetical question, but when we look at in TB, when we drive a T cell strongly with its cognate ligand, it really doesn't do very well in the inflammatory milieu. But if we drive that T cell with a slightly suboptimal peptide, then when it goes into the inflammatory site, it's much more persistent, and it actually behaves better, and it actually mediates protection more for a longer time frame. So the idea is, what is a good T cell ligand? Is it, and of course it depends on the ultimate function of the T cell, but is that something that we should be discussing? I know it's sort of outside what you guys are looking at, because you want that really excellent. The first question is, what's the best signal? And then what's the strongest signal? And then the question is, what's the best signal? Yeah, well, it's an interesting question, because normally we have interest in CD4 responses to tumor-derived antigens, to self-derived antigens. And we're interested in looking at whether actually studying the best binders of a self-antigen might actually be a bad idea, because potentially they might be the ones that might produce the most regulated response. So yeah, it's an interesting question, and we can only help by marrying together the sort of binding of the peptide into the HLA with the immunogenicity data of what we actually see a T cell response to, and try and correlate those kind of things.
Great, thank you. Right, well, so thank you so much, Bruce and Andrea, for two great presentations and a great discussion at the end of that session. So now we're going to go into a 20-minute convenience break, so you've got a chance to quickly go and grab yourself a coffee, and we look forward to, so please stay on the call. You don't have to leave the call. We'll just put the blank thing on the screen for you. Please stay on the call, and we'll see you in about 10 past 3. We'll be back here for the next session 3. Thank you.